So thank you for coming to another seminar session. I think it's the last one of the year. So after this, we will take the summer break and see you again on September. So today, to celebrate this last seminar, we have a special seminar with the people that have the high scholarship. As you know, these high scholarships that are given by the SIC and also in EDEA. And those are Xavi Bachot too. I don't know that they are different calls, but are some SIC scholarships for recently graduated students to start to do some research projects here in the institute. So today we have four of them, the four people that won this scholarship last year, David, Xavi, Patricia, and Blanca. And they will be talking shortly about their research. So they have more or less 10 minutes, and we will leave 2 minutes for questions. And if not, we can have more questions at the end of all the talks. So I don't know who is the first one, David. So thank you very much. And also then making the questions, be nice. They are students, they have done a lot of work. So let's try to be instructive. So thank you very much. Thank you.
Hi everyone, so I'm David Fernández, and my JAE internship consisted of 2 different projects. So I'm going to explain the 2, but I'm going to focus more on the first one. So the first one is related to Posidonia oceanica restoration, and the other one, the second one, is about physiology. So the first one, it's titled Initial Approaches to Posidonia oceanica Rhizome Micropropagation. So as you know, Posidonia oceanica is a seagrass endemic to the Mediterranean Sea that forms extensive meadows and that provide a lot of ecosystem services to societies. However, these meadows undergo a marked decline due to human disturbances, such as anchoring or discharges, basically consequences of demographic growth and mass tourism. And restoration aims to mitigate this degradation of meadows. So currently, restoration of Posidonia oceanica relies on extracting fragments from natural meadows to then do replantation, planting. So that's one of the main limitations that restoration has. So to overcome this, we propose the development of a micropropagation protocol of Posidonia oceanica in order to, from one or several fragments, plants at the beginning, to multiply them in vitro, culturing them, then to create a huge stock that is going to be available for future restorations. So I conducted many experiments, but I'm going to focus on some of them. And the fragments we collected were from the Bay of Alcudia, and we dissected it into explants, this that you see, which is formed by a little piece of rhizome, and then the young leaves with the emeristematic tissue. And we cultured them. So I have also an example here that you can see. Well, this is a solid medium, but you can see the explant is growing green. And we conducted different experiments. So the first one, we studied different salinity levels in culture. So why we tested different salinity levels? Because we also wanted to test the effect of addition of sucrose as a carbon source. And if we added sucrose at the 35 salinity level, PCU units, so we would increase the osmotic pressure of the medium. So we lowered the salinity in order to alleviate that increase in osmotic pressure, since explants, since they are dissected, are more vulnerable to changes in the medium. So comparing these 2 salinity levels, 30 and 35 PS units, we saw that survival was lower when we lowered the salinity, and it was significantly different. And we also studied the health status, measuring greenness, that is a measure, a subjective measure of how green an explant is and how viable it is. And we saw no differences, so that means that it doesn't affect negatively. And sucrose, we tested 4 different concentrations. We tested 0, 5, 15, and 30 grams per liter, and we saw that the addition had no effect on survival and on health status as well. Another experiment we also did was testing different culture media. We tested 4, 3 who were that were commercial mediums for terrestrial plants. They were Morasifian School, Heller, and Hogland medium, and those were solid commercial formulations who were meant for terrestrial plants with lower pH of 5 and with 5.8 and a salinity of 0. So we found out that the suitable culture medium for Posidonia oceanica was the protozoan-rich seawater medium, that it was meant, the target organisms are algae, but it's also meant for marine culture, so higher salinity and more similar pH to the one that Posidonia oceanica needs. We also tested the addition of growth regulators, that those are molecules that have an effect on growth and development of the explant. And we tested anauxin, naphthalene acetic acid, and acetokinin, metatopolin, in different concentrations. We tested combinations of 3 concentrations of naphthalene acetic acid, who were 0, 0.5, and 1. And then 5 concentrations of metatopolin that were 0, 0.25, 1, 2, and 4. And we found out that compared to the control, there was the control is represented in yellow. There was only one treatment that had a better performance regarding survival, who was the combination of 0.5 naphthalene acetic acid and 0.25 metatopolin. So that indicates that concentrations close to those levels may be beneficial, so more research is needed, focusing on these specific hormones, and also that suggests that they work synergistically in those concentrations. So we also tested health status, and there was no differences, as well as leaf growth in length. Leaf growth was observed, but there were no differences among treatments. So the addition of growth regulator was not toxic to the plants. So we have seen that explants remain green, that explants grew, but no division was met at this stage. So we wanted to achieve this as a goal, a final goal of micropropagation. But I think it's a matter of the duration of the study. Like Posidonia oceanica is a very slow growing seagrass, so it takes more time than expected. But this is, however, an important contribution to rhizome micropropagation that moves toward restoration independence for natural mediums. So changing topic, in my second presentation is titled Monitoring the Energy Dynamics During the Reproductive Season of Seabass. And this frames in dynamic energy budget models. Those are models that aim to study how energy fluxes within organisms. So they link food intake, how energy is stored, and its investments in reproduction and in growth. So we studied it in the European seabass, and what I particularly did, I studied 3 different organs: muscles, liver, and gonads, which I processed samples. I dried them and I ground them, and we measured its energy content with a calorimeter, which is this. right here, and results showed that well, some evidence of energy mobilization between the liver towards the gonads in the reproductive season before spawning. So as you can see in this graphic, this represents the gonad size throughout the reproductive season till a maximum when is the spawning of the eggs. And we saw that at the early stages, energy, as you can see, is storage in the liver. It's more energetic, and then energy starts to be mobilized to gonads, who start increasing in size till a maximum at the spawning, when we saw that gonads were much more energetic than the liver because energy has been mobilized. And then after that, gonad size decreases as well as energy, since energy is released in the eggs. So these results are promising and are going to be integrated in the dev model. And that brings us to the end of the presentation. Thank you. Any question?
How do you estimate the energy? I don't know if I missed that explanation.
Okay, so how the calorimeter works is that you went with a ground sample, it burns the sample. So then you get the calories that are in that sample in terms of density, and then you can compare it.
And then you also take into account the total weight, I guess, of each one.
Yes, of course, but in this case, this was more interesting to see because since gonads are bigger than the liver, so you would see always like the gonads are more energetic in total energy content in the fishes we studied.
It's for unit of mass of the organ. No,
Yes, this is a density of energy. So joules per gram.
Interesting.
Mhm.
Any questions for David?
Now that it's over from the first experiment, what would you change if you have to redo it again?
The first of the Posidonia.
Would you change anything, or do you think the methodology was okay and you find everything going? It's not a tricky question.
Yes, I know. Yes, I also tried like if to replicate more salinities and more concentrations, like to see the gradient, because since there was no published bibliography, o sea, references about Posidonia oceanica, we did not know like the concentration we were going to choose. So maybe like trying a broader range of concentrations, but that would imply like to have less replicates. So that would be a compromise of the experiment.
Okay, so thank you very much, David.
Well, good morning, everyone. So my name is Blanca Nieto, and today I'm going to be talking about what I've been working on the past few months within the PDO team and within the JAE program. So I'll be mostly focusing on the topic of my master's thesis, but I'll mention the separate parallel projects that we've been working on. So all in all, this is the light sensing of planktonic diatoms and community movement of benthic diatoms. So I want to first talk about light, start with there. So light is essential for life on Earth, and this is not just for the as a source of energy. course, but as a source of information as well. So this is also the case in the ocean. However, when light enters the ocean, it begins to be absorbed, and the first wavelengths to be absorbed are red and far-red light that can reach, in some occasions, around 2 meters of depth, and blue light being the one that reaches the farthest. So since it's so essential, it's important for living beings to be able to detect it. So they have photoreceptors for this. Now we'll be focusing on the phytochromes that are photoreceptors that are sensitive to red and far-red light in particular, and they are activated with red light and deactivated with far-red light. And of course, the diatoms that have these photoreceptors. So adding this together with the fact that the diatom distribution along the water column is very wide, it's quite puzzling to see why they have the photoreceptor that is sensitive to a light that is not present in most of the habitat that they live in, no? So because of this, there are current hypotheses that have arose to try to explain this. Firstly, it could be that the phytochromes are detecting the light from the sun, so the red and far-red light that is only in the surface, so as a surface sensor. However, it's also possible that they're detecting not the light from the sun, but rather the light that is out of fluorescence from the chlorophyll. So if this is the case, it could be detecting their own plastid status, so their own autofluorescence to sense the status, or it could be detecting the autofluorescence from other neighboring chlorophyll containing cells to know the abundance or the presence of other neighbors. And lastly, more recently, it's been seen that potentially the phytochromes and diatoms are not just sensitive to like red and far-red light, but also to other wavelengths. However, more research is needed to understand if this is the case, if the response is from these other wavelengths or other reasons. So if that were the case, they would be integrating the light spectrum in order to understand their depth in the water column. So in order to shine more light on this topic, we have performed some experiments. So we have exposed the planktonic diatom tricornutum to different wavelengths, so to blue, green, orange, red, far-red, and dark. And we have exposed the wild type and 3 knockouts of the same diatom without the phytochrome, so knockout of the phytochrome, and a recovered. The knockout with the gene of the phytochrome reintroduced as a control for the mutation. So after the exposure, we filter the medium, the culture, in order to obtain the secretome, so the medium with all the metabolites that are secreted during that exposure. And we also conserve the filter for future analysis. So in these experiments, what we'll be analyzing is the secretome, and we'll be analyzing with HPLC mass spectrometry, so high performance liquid chromatography that separates the molecules that are present in the secretome, in order to be identified with the mass spectrometer that gives a specific spectra for each molecule. In that way, we will obtain groups of metabolites that are present for each of the conditions. And the experiments that we'll be analyzing, not yet, so these are the expected results, okay? So we will be analyzing far red, red, blue, and dark. And we'll be analyzing in one wild type and 1 knockout. So with this, the first result that we're expecting is the dark will be the background, so the metabolites that are secreted naturally, not as a response to the stimuli that we're exposing them to, but naturally secreted. Next, the red light will be causing some group of metabolites that are a response to an active phytochrome. Then the far red will be will have a group of metabolites that are with an inactive phytochrome. And then we of course expect to not see these results in the knockout, right? Because it's not sensing, it doesn't have the phytochrome, so it's not supposed to be sensing the red and far red light. So it's the same as if it were in the dark. Next, so these are similar experiments that were performed in another study that they exposed the wild type to red, far red, and dark, and then transcriptomic analysis. So they saw genes that were expressed for the different lights. So that's what we're basing the conclusions on, no? And then what we add is the blue light, no? So we expect to see groups of metabolites that are secreted as a response to the blue light, not relevant to the phytochrome, but just as a response to that stimuli. So they will be present both in the wild type and in the knockout. And furthermore, if it's the case that the blue light is detected, is sensed by the phytochrome, we expect to see the same metabolites that are secreted in the red light. We expect to see them in the blue light. However, that can be the case as a response to the blue light, or it can be the case that the blue light is causing the autofluorescence, so emission of the red and far red light, and that's what is detected by the phytochrome, not the blue light itself. So we can't know this with these experiments, and neither can it be with the other experiments that I mentioned. So that's the main conclusion that we want to that we're expecting, no? But it would be ideal if we are also able to identify specific metabolites that are related to a specific pathway, you know, to understand what changes are caused by the light, you know, so essentially infochemicals that are transforming the light signal to a chemical signal, amplifying it, no? So, well, this is the first part the topic of the master's thesis, no? And I've been talking about how light is perceived by planktonic diatoms. Now we're gonna move to the effect that light has on benthic diatoms, in particular with Semilavis robusta. So it's been seen that the light has an effect in the movement in the first couple millimeters of the sediment as a response to the abundance of light that can change with the tides that cover the sediment, or with clouds, or the time of day, it changes, no? So they move within the sediments. So we wanted to study how this movement is done. So they designed a microchip that represents a two-dimensional, it's a two-dimensional representation of a sediment in which this movement can be tracked, can be explored. So in the first chamber, the idea is to introduce diatoms in the first chamber and cause a stimuli in the second chamber that can be silicates or light, and see the movement throughout the two-dimensional sediment. And that way we can track them and see if there's specific tracks that are followed more by, after one takes it, they leave trails. But this is other experiments that we've seen. They leave trails of metabolites that are can be potentially sensed by other diatoms, and that way they follow the same track, having a community movement. And that's why it's such an optimal movement, no? So that's what we want to explore with this. But firstly, we need to have a highly concentrated population of motile cells. So in order to do this, to have this population, we needed to do a growth and motility curve that will help us work with them in the moment that they are motile, because you can see that the peak of motility is really narrow, and it's really from one day to the other, they start moving a lot more. So this needed to be done previously. And after we can explore the tracks that they take and the community movement that I mentioned. So this is it for me. This is everybody, we're excited. Thank you for listening. Any questions?
Any questions for Blanca?
How are you planning to introduce the light into the second chamber?
Covering the idea is to cover the first one and just introduce it with the light of the microscope. And that really does create a gradient because it's such a small surface that even the shadow of the covering will create a light gradient within it.
Question? Okay, so we can move to the next speaker. Thank you very much.
Hello, everyone. Thanks for being here. My name is Xavi Bachot. I'm a student. I'm a marine ecology of marine ecology masters at the University of Illes Balears. And this project forms the core of both my master thesis and also the internship that I'm having at IMEDEA. And this project is done through the JAE Intro Programme funded by UIB-CSIC. And by under the supervision of Dr. Alejandro Fila and Dr. Ismael Hernández. Today, I'll be presenting the Mediterranean Sea as a trap, flow structures, and accumulation of floating debris. So, moving to the introduction, floating debris, especially plastic, is one of today's most pressing environmental challenges, with over 8 million tons of plastic entering in the ocean per year, affecting more than 700 species. And this plastic is buoyant, is persistent, and can travel long distances. So, but why the Mediterranean is a hotspot for the floating debris? Well, despite covering less than 1% of the ocean surface, it contains nearly more than the 7% of the global microplastic. And this is due to the high coastal pressure, tourism, and shipping. But not everything is anthropogenic. The Mediterranean is a semi-closed basin with limited water exchange that act as a natural trap. On the top of that, talking about ocean dynamics, we have a complex surface circulation with different mesoscale structures like heavies, jets, and fronts. Okay, so here the accumulation of floating debris is not random. Okay, it's followed by the ocean dynamics. Also, we have a strong east-west biogeochemical gradient that influence both biological and our floating debris. Okay, so based on this, we hypothesize that floating marine debris in the Mediterranean tends to accumulate in specific areas due to mesoscale and submesoscale structures that act as a dynamic traps. So how are we going to verify this? First, we are going to identify potential accumulation zones using Lagrangian divergence from high resolution velocity fields. Also, we want to combine the divergence and the chlorophyll A to detect physical biological coupling. And for achieve the first objectives, we want to apply self-organizing maps to classify mesoscale transport patterns. Okay, moving to material methods. To carry out this analysis, we use two high-resolution data sets from the Copernicus Marine Service. The first one is a Mediterranean sea physics reanalysis. What is a reanalysis? A reanalysis is essentially a combination of ocean model simulation, in this case it's NEMO, and real observations that can be satellite data or in situ observation. In this case, it's ocean-bound. Okay. And for the second data set, it's a chlorophyll A concentration. It's a proxy of biomass phytoplankton that was provided of all these sensors, lots of sensors. Okay. The thing is, we have two different resolutions. As you can see, for the chlorophyll, we have 1 kilometer, and for the velocity fields, we have 4 kilometers. So we have to interpolate this grid to the other one in order to combine them and work together. Okay. So, but how are we going to study this transport? Okay, first we want to see how this works, because there are two perspectives. The first one is the Eulerian. Okay, as you can, we observe the flow properties passing through a particular point in the space, and the other one is the Lagrangian perspective that observes the flow properties following fluid particles moving through the flow. Okay, so in our project, instead of looking at a fixed point, we want to track these particles because we want to know where the floating debris or the nutrients or the chlorophyll accumulate. Sorry, not the chlorophyll, the phytoplankton accumulate. So as you can see, we are having filaments, and this is only possible with the Lagrangian perspective. Okay, so here I want to show a comparison between the Eulerian and the Lagrangian. In the Eulerian, it's a bit noisy, it's a more chaotic, but the Lagrangian, it's more precise. We have more accuracy. This is an image of chlorophyll, a satellite image, and I wanted to show you that the Lagrangian perspective fits better with the chlorophyll. Okay. So now I want to explain you what is a sum. A sum is an unsupervised learning algorithm that reduces all this amount of data, this high dimensional data, to a 2D grid while preserving topological relationships. Okay. I'm going to try to explain this. It's a bit difficult because for me it's really difficult. This is all our data. This blue cloud, it's all our data. Okay. This 2D grid, it's the sum. It's not trained yet. Every node of this grid is a common pattern that we can find in all our data. So every time that we feed the sum, we're going to feed with an input of velocity field. So its input is a velocity field, as I said, and the map selects the closest node, the pattern that is closer. So with more inputs, nervy nodes are just together. So the result is a map of typical surface flow patterns. Don't worry, now I'm going to show you. So before moving to the results, I wanted to mention that we have a problem about temporal resolution. Okay, because as I said in the material method, we have daily data and monthly data, but the Lagrangian divergence relies on following the particles. But if we only have data month by month, we are not following. We are losing information. Maybe from since the first month to the last month, it's happening lots of things, lots of structures that we are missing. So we have access to daily data, but we're still working on it. That's why I don't want to show you incomplete work. Okay. So, but in any case, we work with monthly data and we had interesting results. Okay, so now I'm going to start with the results. This is the first sum that we did. This is, as you can see, there are 9 patterns. These 9 patterns resume all my data of since 1998 to 2022, I think. And as you can see, every pattern has a percentage. So what does it mean? Well, this is a seasonal frequency. As you can see, there are different colors. The darker colors has more dominance. So with this, we can know how often every pattern appears during a year. So as you can see, for example, the P9, the darker color, it's November. So I go back. So for the P1, this is probably P1, it's November, so November it's winter, no? Yes, no, sorry, autumn, sorry. So this is the common pattern for autumn. Okay, so that's really interesting. But the real interesting is this. This is our objective. We wanted to pass from the circulation, from the velocity fields, to the surface aggregation, to the Lagrangian divergence. In these figures, the positive values in red reflects dispersion, reflects divergence. But in blue colors, we have the convergent zone. It's really interesting. I can see which part of the Mediterranean has more convergence, more divergence. But the thing is how we can prove that. Because okay, we have this, but how we can prove that? So we were thinking about the chlorophyll, because if you find a convergent zone, not only the plastics is going to converge, maybe the nutrients, maybe the phytoplankton. So we wanted to work with chlorophyll. So the first thing we had to do was to compute the velocity field to the Lagrangian divergence, and then we couple and we train another sum. The thing is, the results that we had only represents the coastal apport, the coastal chlorophyll. And we want to know the mesoscale structures. We want to know what's happening here, what's happening here. Okay, so we were thinking, okay, can we mask this? Yeah, of course, we masked it. We took all these patterns and we masked them into this. Now we have all the parts that we don't want to have, and we can observe different mesoscale structures. This is they go together, okay? There's a couple. So as you can see, between the islands of Corsica and Sardinia, we have something interesting. Also in the Strait of Sicily. Those are my first results, our first results, and we're still working on it. I cannot say you really anything is interesting because this is monthly data, and we want to work with daily data to have more precise results. Okay. Also, we did the seasonal frequency, and as you can see, we have two mark seasons. In this case, is the summer for P1. As you can see, the P1 represents 36.3% of all our data. It's a lot of this. And also the P8 that represents the winter, more or less. It's interesting. The discussion and the conclusion is still working in progress. And that's all. I hope that you enjoyed. If you have any questions.
Thank you very much. Very interesting. Any questions?
Yeah, actually, I was confused with the self-organizing maps.
Yeah.
So the 9 plots that you put together,
Yeah.
these are the 9 dimensions of your self-organizing.
Exactly. This is not a. It is a 4x4. In my case, it's a 3x3. That's it. You can train whatever you want. So this is like the difference between the other one. It's a bit difficult to explain, right?
So each of them explains somehow part of your variance of your data.
Exactly.
Depending on
It
which
takes
community you're in the year.
exactly. It resumes like all my data in 9 patterns. So if input of data it's close to any of these, so it's increasing the percentage of each one. It's like the idea. This is sorry, I can't explain that.
And why there are 9?
So because it's, with 9 it's enough. Like if you have 4, it's 16. It's really hard to explain your data, no? It's also if you have like only 4, like 2x2, it's you are short of data. So 3x3 is like the perfect combination.
Any other question?
I didn't understand the masking thing of the profile on the.
Okay.
What do you mean by mask?
Mask, as you can see, we have the river discharge, human coastal pressure, etc. But we don't want this. That's why we took these results, okay, these patches, all of them, from the pattern 1, 4, 5, and 7 that you cannot see because if not, you are not going to see anything. We took it, and for the next sum in the phase 3, we generated another sum, and we masked it from the initial data. So now we are not having this data. As you can see that here we had this. Now we are not having here. Also here that we have all this river discharge, we are not having. So that's interesting because the machine learning it's helping us to generate information that we couldn't see in the beginning. Now we can see some patches that in the beginning we couldn't.
So it's like you change the scale of the maximum and minimum, so you omit the maximums that are in the coastal and.
No, not the maximum. So we took literally this point of data and we removed for our initial data.
Okay.
we try to do the maximum and minimum, but the thing is, probably here you don't have a maximum, but it's a river discharge.
Yeah.
So if we wanted to take it out.
Have you tried using the logarithm of chlorophyll instead of the?
No.
Is that what normal?
Can be on the. No, we didn't. We should try this.
Now that the question arose, what is chlorophyll, I really start to understand this. So it doesn't mean you have a lot of data for the Mediterranean on the right map, but you just chose the white color. Because the white color for me feels like there's no data.
Oh, maybe there is data, but the maximum of chlorophyll it's the relative.
But by choosing another color map or by doing a logarithmic scale or something, could you make this visible, the other one?
It can be possible, but I didn't try it yet.
But so you have data, but you just don't know what's it's close
Yeah.
to zero. But
Probably.
it's a satellite image, right? It's a satellite image.
Yeah, see,
Okay, so there might be something measured, but in a very, very small.
exactly.
Okay.
Because this is a sum, it's a machine learning. Probably it's masking the chlorophyll patches that we have here, but because the number it's really low, this is higher, so that's why.
Okay, thank you very much.
Hola, bueno, me llamo Patricia y os voy a hablar pues del rol que juega Posidonia oceanica en el secuestro de carbono azul y cómo se relaciona con la arribación de las playas. Bien, durante la beca he trabajado principalmente en dos líneas de investigación, una dedicada a la medición de gases de efecto invernadero en Posidonia oceanica, en la arribación de Posidonia oceanica, y otra en el secuestro de carbono azul en praderas marinas. Quedaros con esta imagen porque posteriormente mencionaré igual que esto es un llamado cual que también lo voy a mencionar ahora. Bien, antes que nada, ¿cómo se forma la arribación? Bueno, depende también del ciclo anual de Posidonia oceanica. Y bueno, la imagen es bastante gráfica y podemos ver cómo, dependiendo del oleaje, se forma o se desmantela el arribación. Un oleaje fuerte transporta la mata muerta y se queda depositada en la línea de las costas, por lo tanto se forma el arribación. Y este puede desmantelarse, volver más hacia adentro o redistribuirse con oleaje más débil, que suele pasar en verano y en primavera. Bien, para llevar a cabo este estudio lo que hemos hecho es utilizar un analizador portátil llamado LICOR, que mide los flujos de CO2, de metano y de vapor de agua. Donde se ha realizado el estudio ha sido Calamillor y el Puerto de Canonge, una zona urbanizada y una zona natural. Lo que hacíamos, pues realizábamos incubaciones cerradas, colocando la cámara, que es esta, sobre distintos tipos de arribación, seco, húmedo, arena y artificial accumulation, que es la imagen, que es esto. Bien, estos son unos de los resultados que hemos obtenido. Podemos ver cómo hay diferencias estacionales debido al aumento de temperatura, mayor aumento de temperatura, mayor emisión de gases de efecto invernadero. Podemos ver además cómo el arribazón seco, debido a las altas temperaturas que pueden alcanzar hasta 50, emiten más gases de efecto invernadero, entre ellos el metano. Los siguientes resultados muestran diferencias como intentamos comparar Calamillor con el Puerto de Canonge. Sin embargo, podemos observar que los datos son prácticamente parecidos y la única excepción es el montículo, ya que es un mix de materia orgánica, de plantas, de algas que arrastran con la excavadora y lo depositan, ya que a los turistas les estorba. Bien. Como conclusión, todavía no hay una conclusión clara porque este estudio todavía se está llevando a cabo y se siguen analizando datos y variables, pero sí podemos ver cómo un aumento de temperatura está relacionado con un aumento de CO2, de emisiones de gases de efecto invernadero. Bien, la siguiente línea de investigación a la que me he dedicado ha sido el secuestro de carbono azul en Posidonia oceanica. Sabemos que Posidonia oceanica juega un papel súper importante como sumidero de carbono y además es capaz de acumular carbono orgánico circundante y de la propia planta y quedar enterrado en el sedimento durante décadas. Por lo tanto, es una gran reguladora del ciclo de carbono. Se estima que las praderas marinas pueden crecer 2 milímetros por año, por lo tanto, nuestras muestras son 30 centímetros, aproximadamente tenemos datos de 150 años. ¿Cómo hemos llevado a cabo este estudio? Bueno, utilizando los llamados cores. Son tubos de PVC, estos son tubos de PVC que se insertan sobre el fondo marino, se clavan y extraemos una columna de sedimento. Esa columna de sedimento representa pues, un archivo natural de la historia de la acumulación de la pradera. ¿Y qué podemos obtener con esa columna de sedimento? Bueno, pues múltiple información como materia orgánica, carbono inorgánico, ADN ambiental, nitrógeno, además de episodios de enterramiento, cambios ambientales, etc. Bien, para llevar a cabo el estudio, pues lo que hemos hecho es cortar longitudinalmente el core, tomar muestras de cada centímetro, pesarlas, obtenemos el peso húmedo, luego lo metemos en una estufa a 60 grados, obtenemos el peso seco. Posteriormente metemos las muestras en una mufla de 550 grados, eliminamos el porcentaje de materia orgánica y finalmente la metemos en una mufla de 1000 grados y eliminamos ya el porcentaje de materia inorgánica. Y a partir pues, de estos datos podemos obtener los siguientes resultados. También mencionar dónde se han tomado estos cores, y en concreto en Formentera, en el Puyol, Illetes y Calatorreta. Bien, mis datos son los que se muestran aquí, los del año 2024, y lo hemos comparado con otros estudios. Podemos ver, si comparamos mis datos con los de 2012, en la misma ubicación, vemos como hay diferencias claras. Mis datos son más homogéneos, en torno al 2%, y sin embargo el de 2012 pues, presenta una mayor variabilidad y mayor porcentaje de carbono orgánico. ¿A qué se puede deber esto? Bueno, en 2020 hubo una tormenta que arrasó con buena parte de Comunidad Valenciana, Baleares y Cataluña. Por lo tanto, se cree que se ha erosionado buena parte del sedimento. Por lo tanto, mis datos, en concreto de Illete, no representan el verdadero carbono orgánico que debería de haber si no se hubiese producido la tormenta. Bien, Calatorreta, mencionar que estos cores son decimooceanodosos y lo hemos comparado con otro estudio en Creta, simplemente para que os hagáis una idea de los valores en los que oscila el enterramiento de carbono orgánico decimooceanodoso, que podemos apreciar que es menor que Posidonia oceanica. Bien, Puyol sí muestra un perfil relativamente parecido, y esto se debe a que en el Puyol nos encontramos con un gran arrecife de Posidonia oceanica. Aquí podemos ver una pared de Posidonia y bueno, aquí hay una persona para que os hagáis más o menos una idea de las dimensiones de dicha pared. Por lo tanto, los valores son más altos debido a la grandísima cantidad de materia orgánica que hay aquí. Bien, también hemos podido calcular los hechos de carbono. Para ello, nuevamente, hemos comparado con la bibliografía y bueno, podemos ver cómo el Puyol muestra valores similares en torno a 7 kilogramos por metro cuadrado, parecido al año 2012, y es Illete, pues sin embargo sí hay diferencias por lo comentado anteriormente. Y bueno, pues Calatorreta tiene más hechos de carbono comparado con comparado con las muestras de Creta. Como conclusión, bueno, sabemos el papel tan importante que juega Posidonia oceanica, es capaz de acumular carbono a lo largo de las décadas, incluso se puede hablar de siglos, pero sin embargo cuando esta Posidonia llega en forma de mata muerta a las playas, se forma la ribazón y es capaz de emitir gases de efecto invernadero. Sin embargo, es capaz de capturar más carbono viva que lo que emite muerta. Por lo tanto, el siguiente paso será cuantificar el balance entre captura versus emisiones.
Muchas gracias, Patricia. ¿Una pregunta?
¿Tienes una idea de cómo está afectando el aumento de temperatura a la Posidonia? Porque hace un tiempo ya veo que era en 18 grados moría, Y ahora también leía que tuvo que dar más
Eso no lo hemos estudiado con estos cores, no son uno de los parámetros que buscamos.
Hice un experimento y moría a partir de los...
¿Del 4?
Sí, no sé si pensamos que puede ser una detección de clones. Pues igual los clones los clones que no pueden resistir a la temperatura han muerto, entonces los que se han quedado no. Pero a mí a partir de los 34 se me y estuvieron semana y media.
Me parece curioso en las emisiones, o sea, de carbono, aquí, el anterior, sí, que la diferencia entre la mata húmeda y seca, que en primavera la seca emite mucho más, pero luego en verano la húmeda emite. Estas diferencias les dan una explicación.
Todavía no te podría dar una explicación, pero bueno, la mata seca en algunas ocasiones pues emite más porque alcanza mayores temperaturas. Y bueno, pues la húmeda oscila entre los 30 grados y pues algunas veces se formará un microclima que aumentará la emisión de gas.
¿Alguna pregunta más?
En función, no sé si está muy relacionado o no, pero en función a tus resultados que has visto al final de los arribazones versus las en sí, ¿recomendarías que se quitasen los arribazones de las playas?
No por el papel que juega, frena la erosión, además que el hábitat pues, de muchos invertebrados, insectos, entonces bueno. Y al fin y al cabo, lo que dije en la conclusión, absorbe más carbono del que emite muerta.
Que avienta turistas.
A lo mejor en primavera igual se pueden mojar. Para que evitar menos. Vale, pues lo dejamos aquí. Un aplauso a todos los ponentes.